This video demonstrates an approved workflow of the Playbook Workflow Builder. Use Case 5 can be used to discover small molecules that may be capable of inducing specific biological processes in cells. In this video, we use autophagy as the target biological process that we aim to induce. It is important to note that the analysis presented here is only intended to communicate ideas for basic biomedical research. You should not consider taking any of these drugs without a prescription. Autophagy involves the degradation and recycling of proteins within a cell, and for some diseases, such as Alzheimer's disease, induction of cellular autophagy is expected to be beneficial. The workflow utilizes various phenotype databases to obtain sets of genes associated with autophagy. Subsequently, the Links L1000 chemical perturbagens library is employed to rank potential drugs and small molecules that are found to influence the expression levels of these autophagy-associated genes. You can input the biological process of interest to start. The default input is set to autophagy. Next, we query Enrichr with the term autophagy to find matching gene sets annotated with this term. From the Enrichr term search results, we extract gene sets from KEGG pathways, MGI mammalian phenotype, and gene ontology biological process libraries. Return gene sets are associated with the desired biological process. After extracting the matching gene sets for our input term, we convert the gene sets into the GMT format. GMT stands for gene matrix transposed, and it is a common format to store collections of gene sets. From the GMT file, we combine the gene sets into one gene set using the union set operation. A gene set from the geobiological process contains 347 genes, the MGI mammalian phenotype contains 56 genes, and the KEGG pathway contains 137 genes. After computing the union gene set, we query the Links L1000 database utilizing the SIGCOMM Links API. It identifies drugs and small molecules that may upregulate the collective expression of the genes in the set. SIGCOMM Links is a web server application developed by the Mayan Lab. It serves over 1 million human gene expression signatures for search and download. Accordingly, we retrieve the top drugs and small molecules based on their corresponding Z-score, which signifies the extent of their potential influence on the input genes. As a result, a consensus-ranked list of drugs and small molecule candidates predicted for inducing the targeted biological process is produced. We compute the mean score value of the 3 resolved drugs from each database: MGI mammalian phenotypes, KEGG pathways, and geobiological processes. The scored value of a drug signifies how effectively it mimics a specific signature. Lastly, PubChem API filters the ranked consensus drugs, retaining only FDA-approved drugs. The workflow returns the list of 67 drugs most likely to be effective in inducing autophagy. Baricitinib has been returned with the largest negative Z-score value, and there was a study by Celia et al. In a paper published in Cells demonstrating baricitinib has been speculated the additional capacity to interfere with the autophagy pathway. The author mentioned that larger studies are needed to clarify this aspect. Again, it is important to note that the analysis presented here is only intended to communicate ideas for basic biomedical research. You should not consider taking any of these drugs without a prescription. As an overview of the workflow, it starts by assembling a collection of gene sets associated with pathways and biological processes related to our target phenotype. To achieve this, we utilize the Enrichr's metadata search feature. The gene sets are identified by Enrichr from the 3 databases. Collected gene sets are converted into a gene set library and saved in the GMT format. We compute a union gene set from all these gene sets. We submit the union gene set to SIGCOMM Links using the SIGCOMM Links API to identify Links L1000 chemical perturbagens that maximally upregulate the collective expression of these genes. Once we have a set of ranked drugs and small molecules from SIGCOMM Links, we calculate their mean scores. Finally, we filter small molecules with PubChem API to keep only FDA-approved drugs, and the final set of ranked drugs will be displayed in a table. With that, I'd like to acknowledge the members of the Playbook Partnership team from xRNA, Glygen, Kids First, Links, and Metabolomics Workbench for all of their work over the past year. I'd also like to thank the CFDE community and our NIH program officers for their continued support. Thank you all for your attention.